All right. The first thing to know about the lab leak conspiracy, or the lab leak idea, if you want to be open minded about the thing, is that when you go to coronavirus conferences, and I've been to two since the start of SARS-CoV-2, COVID 19, this doesn't even come up. It doesn't rise to the level of something that people even consider worth entertaining as an outside thought. Because essentially, it doesn't make any sense if you understand viruses and if you understand coronaviruses particularly. Now, the thing you'll notice about, the thing you could notice about the people that push the conspiracy the most is that none of them are virologists, and they all come from some field other than virology from which they probably haven't read a lot of virus papers. And although I hate to say it, kind of don't know what they're talking about. So, let me walk you through. And I guess I haven't said which side I'm going to come down on, but you'll find out in a minute. Here are some of the papers that the people who have been sort of promoting this lab leak idea probably should have read and considered. And yeah, these are at least the things that are helping to form my opinion. I think the important thing before we start, though, is to say that scientists, being a scientist is a job you do. You put on the lab coat. You are not born with it. Nobody is a scientist all the time. It is trying to be careful. It is trying to not make mistakes. And so if you have that metaphorical lab coat on and somebody asks you, was the virus from a lab leak? The most correct thing to say is that it seems very unlikely, but, yeah, I guess I can't rule out any possibility because I wasn't there. And that's correct. That's how you don't make a mistake. But it doesn't mean that anybody actually thinks that. There's poll after poll of virologists, and yeah, they do not, especially the ones who actually know what they're doing. Like, it's kind of inversely proportionate to how well you understand coronaviruses is how much you believe in the lab leak idea. Yeah, so there, maybe I gave that away. Here's a really nice molecular sort of drawing of SARS-CoV-2. The only problem is I think the M proteins, which are these little pink blobs with a yellow bit on there, I think those are still a little too disorganized and maybe a little far apart, but they're pretty good. They're a whole lot better than you get on any of the other models. And they cracked it open so you can see a little bit of this stuff inside. And that's going to be where the genome is. So, for a virologist, a virus is its genome. So there would be no way to tell from the amount of smog over a city, or somebody giving you a shifty look, or somebody that you don't like but might be up to something. There's no way to tell if that has anything to do with a virus, and no serious scientist that I know of would actually even consider it. The way you detect a virus is by detecting its genome, because we have tools for that. So one of those is a technique called PCR, polymerase chain reaction, where you can take, if you know two little parts of it, and we know the genome of this thing, you can grab onto those and you can copy whatever's in between. And then there's another technique called sequencing, where you can read out that unknown sequence that's in between the two things you did know. And sure enough, if all of that matches, including the unknown part to SARS-CoV-2, then you maybe got yourself a SARS-CoV-2. Yeah. And you do that for the entire genome. That's the way it's done. But essentially, a lot of the evidence, like the evidence you'll see coming up in FBI reports, or the Lawrence Livermore lab threw its hat into the ring on this for some reason, they are almost entirely based on what I would call intelligence, which is stuff like spies do. And spies are maybe pretty good at understanding what people are up to and following people around. I don't know of a better way to do what they do, and I don't actually know all of what they do. But we're talking about viruses here that are actually smaller than a wavelength of light, so light just misses them, and they're too small to see. You cannot tell by what anyone does or how they feel if they are actually sick. It's impossible. Can't be done. You can't tell if they've got a virus inside them and are not yet sick. You just can't tell. And, yeah, here's more. You can't tell when they do get sick, whether it's a virus, bacterium, or, one of there something like 200 different groups of viruses that can infect humans, which is a lot. And there are a ton of them that cause pretty much the same symptoms you would get for SARS-CoV-2. Yeah. Both early and late in infection. There's no good way to tell except by copying the genome. So if the genome is there, the virus is there. If the genome's not there, the virus is not there. And if you hear anybody say, well, I have lots of genome inside me, I keep testing positive, but I feel fine, so I'm going to the Taylor Swift concert, then they're rationalizing a terrible thing that they're going through, but they're also making a pretty big mistake because, yeah, if the genome is in you, then you are sick because you can swab healthy people and the genome is not there. It's not part of our DNA. Doesn't secretly hide anywhere that we know of, out of detection. It's either there or not there, and you detect it molecularly by its genome. The genome is also like a little clock, and that, I think, is the neatest thing about this. There is a very, very predictable evolutionary rate for SARS-CoV-2, and actually for just about every virus out there. Yeah. So here we have time on this axis, and here we have the number of mutations on this axis. And all these little dots are every single strain of SARS-CoV-2 that a human has caught since there have been humans catching SARS-CoV-2. We got the first sequence reported right at the beginning of January, I think January 8th or something. I heard about it. Might have been a couple days before that. And that came from a sample from late 2019, so that's why it's COVID-19 and not COVID-20. Yeah. Here you can see that the virus actually is quite predictable. And it makes about 30 substitutions that stay in the genome per year, which means that a year from now, we're on about 130 substitutions right now. A year from now, it will be on about 150 or 160, substitutions. Yeah, where's my math? And it means you can count back, and there are all these ideas that the virus was secretly here. Years, months, whatever. Your, aunt or uncle had a really bad something that felt just like COVID later, and they were super sick in early 2019. And all you can say from that is that if it was, that one left no descendants. That line went completely extinct. And all we have, the only genetic sequences that we have and that we've recovered from all of time fit on this nice little line. The virus is very predictable. Now, the other important thing is okay, so if this didn't come from whatever, some lab, somebody dropping a test, I don't know what, they're making stuff up. I can make up stuff. The nearest strain that we have now still is this one called RATG13. I think it's from Rhinolophus affinis, which is a little bat, just an adorable little fluff ball, tiny bat. Yeah, microbat. Yeah. And that one has 1,135 substitutions compared to the earliest known strain of SARS-CoV-2. Compared to a more modern strain, it would be 1135 plus about 130, essentially, because it's not changing in a direction toward this thing. It's moving on. Change is unpredictable. It's just that the virus will change. That's all you can ever say. And so that in virus time is about 38 years. Yeah. And the Wuhan Institute for Virology wasn't open for 38 years before this. It was open like 2 years or something before, SARS-CoV-2, or 3 years, something like that. So there's no way. It's not humanly possible. It's not virusly possible, I guess, more to the point. And so If you were to find the actual strain somehow, and I believe it would take a time machine at this point, the strain would look like this, but it would have mutations that you don't see on this side. Other mutations that have since been mutated out on its way to becoming, patient zero, right down here, the first one to jump into humans. And the closest we have are samples from the Wuhan, it's the Huanan market. It's like an animal market where you can buy live animals and seafood. And there are samples there that have both raccoon dog DNA, and that's an animal that we know does get infected with SARS-CoV-1, almost certainly with SARS-CoV-2 as well. And it has a very early strain of the virus, but it's about the same as the early human strain. So we're not sure if the raccoon dogs spread it to the first human, and that's why they're similar, or if the raccoon dogs were infected by the first human that was infected from some other animal in that market, but that's really what it looks like. So if there ever is an actual smoking gun, that's how you'll recognize it. You will look at the mutations, and you will see a particular number, and that'll tell you how much older it is than the particular strain of SARS-CoV-2 that first infected humans. Okay, yeah. Next point, epidemiology. What about this animal market, you say? Well, look, here's a little red dot. That's where the animal market is. Here is the location where people lived who had all the first cases as reported to the WHO. And these are all the people who had some link, who had traveled to the market, who worked at the market, stuff like that. And darn if they aren't all centered around the market. It looks like people do not very much come across the river to use that particular market. Maybe there's a market on their side of the river. There probably is. I have no idea. Haven't ever been there. This is sort of a bigger picture with all the cases that are not directly linked to the market, and yet they are clustered around the market. This is Epidemiology 101. This is actually from an epidemiology sort of textbook. This is the famous mapping of the cholera outbreak by John Snow in 1854. There were all these people getting sick from cholera. They knew, like we know that cholera is a waterborne illness. I don't think they knew that it was caused by bacteria specifically, but whatever. Yeah, we got that now. And so to figure out, like, all right, where is this coming from? What do we do to stop this? He actually mapped all the cases where all these people lived. And darn if they don't form a cluster. And what's in the very dead center of that cluster but a water pump? And so he gets the authority. He removes the handle from the water pump, that particular one, so nobody can use it. There are still water pumps all over the place in other places. And the cholera outbreak goes away because it was probably that water pump. Yeah. And you can tell because there's a cluster of cases around it. So here's later data from February. By this point, it started to spread around the world. It's already in California, stuff like that, I think, at least by the end of February. And here you can see still the cluster is mostly right around that animal market. Now, by comparison, the idea, what if it came out of smokestack of the Wuhan Institute for Virology? Yeah. What if it was a little vial that somebody dropped on their way to their car, that night? The institute is somewhere right down here, and it's in an area where there are never any cases, and there are people that live around that, I believe, but those are not the ones that, because all the cases are centered around this animal market. So that's why that and the raccoon dog DNA / early SARS sample from the floor of the market, those are both real good evidence that there was an animal infection at around the same time as the first human infection, and that's correlation rather than causation, but it's pretty strong correlation, I would say. All right, the next part is a little more technical. So there's an idea, particularly by a person called Alina Chan, and there are a lot of people that are like getting behind her and pushing her and saying, "Yeah, do it. Say the stuff." And she's fine. She's a geneticist. Never worked on a virus in her life, as far as I'm aware. But her idea, and she's been featured in the New York Times, yeah, multiple times with big graphics and everything. Yeah, they really like her there. Her idea, at least the sort of key, is that you can tell that this virus, SARS-CoV-2, was manipulated by humans who actually changed it so that it would become more dangerous to humans, because you had to change it or else it wouldn't even be able to grow in a human. And this does get some things right. People have looked at a lot of bat viruses and they've tried to grow them. And as far as I'm aware, there's only one bat coronavirus related to SARS that has been grown. And that was from a swab. So they are actually getting like probably infected cells off the inside of the poor bat's rectum. Yeah. I'm sorry, poor bat. But most of the time, if you think about it, what happens when, hamburgers and stuff go through your digestive system? Oh, there's enzymes and bile salts and all kinds of stuff. Yeah, they get digested. Yeah. And coronaviruses probably also get, for the most part, digested. At least they would probably lose all of their spikes. They would lose some of the things that they need that would help them to get into the next cell. So anyway, this intrepid group decided to say, well, all right, if the furin cleavage site was put in there by the hand of man to make this thing, to convert it from a non-infectious virus to an infectious one, we should be able to take it out and see what the effect is. And here's the effect. In this cell line, there is no effect. The blue is missing the furin cleavage site. The black has the furin cleavage site. Not really any difference. Here is a different cell line, and in this one, the one where they knock out the furin cleavage site actually grows a little better. So the idea that somebody took a bat virus that doesn't grow in human cells, it grows fine, added a furin cleavage site to make it grows worse or the same, and that somehow started everything is... It's not the stupidest idea. And if you made a list of 30 just off the wall, let's not really think about it kind of ideas for stuff to test out with a new virus, maybe this is one. If you're like, we can't get this thing to grow, let's try everything. Maybe that's an idea, but it's not a very smart one. And it turned out to be nonsense. About half of the coronaviruses that we know have furin cleavage in their spikes. It's like a little activator, but it's not necessary. There's a more important activator that's located a little bit further down into the spike, and it looks as though the furin cleavage, I don't know, it may help a tiny bit in some situations, but it's really not necessary at all. Yeah. And also, it's tiny. Here they took out 4 amino acids. They didn't have to take out the first one, and they didn't have to take out the fourth one. They could have just changed those 2 in the middle, and that would have knocked out the cleavage site. And because it's only 2 amino acids, that means the virus can change from whatever it is now to having or not having a furin cleavage site with about 2 mutations minimum. Yeah. And our virus is an evolution engine. It changes all the time. It changes for a living. So yeah, it's made those changes, and the fact that we don't see it in the world today tells you that it probably wasn't any better than the version that's circulating around your family and mine. Okay, next point. There are several SARS-like coronaviruses that grow fine in human cells and do not have a furin cleavage site. So that's why the whole furin thing is just bananas. Yeah. Rather, you would think that if you learned up to maybe the early 2000s, and if your knowledge base is kind of any more recent than maybe 2010, I don't see why you would still believe this. We've been learning things as we go along, and yeah. So when you get somebody who's hand on heart, dead certain about this, I think it just shows the point at which they or the people that taught them stopped taking in new ideas, essentially. Yeah, which you're only a scientist to the extent that you are up to date, and scientific knowledge cools off real fast, real fast. Yeah, it is a challenge. Yeah, it is a challenge. Um, Anyway, here's several of the viruses. They start with a low amount, time goes on, they get to a high amount. Those are the viruses growing in a variety of human cells, or these are monkey cells. These viro cells are actually really good for growing SARS-CoV-2, although there are no reported green monkey infections that I've heard of anyway. And I wish the green monkeys well. I hope they're fine. Yeah. Yeah, here you go. These are two that were isolated from pangolin. One's reconstructed, and the other one was actually an isolate, I believe, on the top. And yeah, there you go. So all of these have in common that they don't have a furin cleavage site, and they grow in human cells fine. Yeah. There is also SARS coronavirus, the original, and it doesn't have a furin cleavage site, and it grows in human cells fine, and it kills something like 1 out of every 10 people that it infects. That's a horrible thing, and thankfully it is gone for now, but there's enough very similar viruses out there in bats that I don't ever think these viruses are going to be gone for long, unfortunately. They will spill back into the human population as long as there are both humans and bats on this planet. Yeah. That's just the world we live in. There you go. Yeah, okay. So, the virus that was found in the animal market, I believe, was 6 mutations different from the one that first infected humans. So it's real close in terms of time. We're talking about weeks there. There's an idea that we actually know where pretty much every virus came from except for SARS-CoV-2. And unfortunately, that's not even close to true. These are some famous virus infections, and these are the nearest animal isolate that we were able to get. And this is basically considered the equivalent of the smoking gun, but you'll see there's no gun and it's not smoking. Yeah, yeah. So there's SARS, that's the RATG, and closest to the original strain, but it's still about 38 years or 1,135 mutations different. So yeah, not that close. We don't have the original for SARS-CoV-1. You will have heard that it came from bats to Himalayan palm civets to humans. The bat viruses are more different than this. The palm civet virus, the one that is closest to the early strain and actually comes from around that time, is still 108 mutations different, which means even though that was contemporary or contemporaneous with the first human infection that we know of, there's still about 3 years of evolution apart. So this is a weird cousin to the one that actually started it. And yeah, that one is almost certainly gone. Viruses turn over, they change. You can identify roughly when a virus is from based on this change, but yeah, you can't really go back because they don't stick around. They just keep changing. Same thing for MERS coronavirus. You've heard that it comes from bats to camels to humans, and the nearest camel virus to the nearest human virus is, at least that I could find, is this one from Jeddah, but that's still 2 years of evolution different, and that's from like close contact case area. It seems like it ought to be really similar, and yet it is about 2 years of evolution different from the human version. And here's the nearest bat version, which is not that close, 153 years. Yeah, but that's considered reasonable in terms of scientific evidence because there isn't anything closer. And if we find something closer, then we will update this. Science is always being updated. It doesn't stand still for you, me, or anybody. Yeah, even when we need a break. Yeah. Compare that to Ebola virus in West Africa, 2014, famous outbreak, killed tens of thousands of people over there. Really bad. Yeah, kind of shut the world down for a while. We have no idea which animal had this virus before it spilled over into humans. There's an idea that it may have been one of 2 different kinds of bats. There's a phantom sequence that somebody said they had of a little part of the virus in a news story, and then, as far as I can tell, never posted that part of the virus online in any way that anyone else can check. So who knows? But yeah, the nearest one we've got from that area is actually considered a different species, and it's 8,549 mutations, which is almost 430 years of evolution different, which means we got some gaps. But I hear you saying, well, why don't we go sample some more? Yeah. Well, how many bats did you swab, today? Yeah. How many bats did you swab this week, this year? Yeah. Me either. I don't swab bats for a living, but there are a lot of bats out there. Something like 30-40% of all mammal species are different kinds of bats. And so we lump them together and say, oh, bats are a thing. And they kind of are, but there are a lot of different bats out there, and they do not get sampled very much. For one thing, they're awake when we're asleep for the most part. For another thing, they're flappy and a little hard to catch. You gotta, rig up your bat nets. And who's funding, bat catching research? Not that many groups. So yeah, it's a question that probably could be answered better, at least close to the time. But now it is debatable whether you would ever be able to answer some of these questions. Was it a normal strain? Was it a chance event like a recombination? It's real hard to reconstruct that, and the best conclusions have pretty big error bars on them. Okay, last and dumbest thing is the idea that a virus needs human help to spread at all. I mean, consider the logic here. The idea is that, oh yeah, the virus was fine in a bat, but it needed somebody to tinker with it and do something in order to get it in the first human. Once it was there, every single bit of spreading since then has been just entirely the virus is doing. It was just that one part the virus couldn't do. Says who? Viruses infect things for a living. It's literally the only thing they do. It is their job. It is all of their job, and viruses have never needed your help, and they don't need your help now. They're fine on their own. Yeah, that's kind of silly. The only other thing I would say is that the idea of the lab leak is harmful to scientists because it makes it difficult to do research on these things. And it's the research on viruses related to this that allowed for there to be such a fast turnaround to the vaccine, to some of the different therapeutics. Most of the antibodies that were being used as monoclonal therapies already existed. A lot of those already existed at the start of this, or we could come up with it really fast because you have to have all these other ducks in a row, essentially. You have to be able to make large amounts of protein, which is not as easy as you think, because there are some tricks, and those have all been figured out by virologists and structural biologists and people that make proteins for a living, and RNA vaccine people. And yeah, that's how we were able to do anything about it. The idea that you need to clamp down on virologists because somebody somewhere that doesn't know what they're talking about is worried that something that probably didn't happen might have happened, that's pretty bananas, I would say. It's kind of like the equivalent of, think of the last fire you saw, the last structure fire. What was there? Was it firemen in a big, cool, awesome truck? Maybe not red. I don't know if they do them red anymore. Yeah. Should we ban firemen? Should we regulate firemen? Should we make sure that firemen have to be extra trained and certified? And should we make sure that maybe politicians need to sign off on, any time that firemen are called out to fight a fire? Would that make the world a better place? Yeah. Ask yourself that. Yeah. I don't think it would. Essentially, it hurts people like me. Yeah. And that's why it annoys me. So there you go. Yeah. So that's our little thing. Maybe this is like a guide for how you can try to talk about the lab leak idea. Because the problem is, as you've seen, the answer is pretty technical, and that's a bar that people would have to get over. And the conspiracies are really non-technical, and so they're easy for anybody to talk about. And so that's kind of what you're up against. That's the burden of knowing what you're doing. Yeah. So hopefully you do now. All right. Thanks a lot.